All right, we are on our last learning target for our last section for learning target 2, which is development. Timing and coordination of specific events are necessary for the normal development of an organism, and these events are regulated by a variety of mechanisms. So let's take a look at those mechanisms. There will be 3 videos for this. This part 1 is differentiation. Individual cells are able to carry out the functions of life, but they can also join to form more complex unities, or entities, sorry. So we know that because all living things are made up of cells. If they are not made up of cells, one or more, they are not considered alive. So that means that even the smallest single unicellular organism can carry out all the functions of life, but when we combine those together, they're able to form much more complex things. So here's kind of this level, what we call the levels of organization, from the smallest unit to the largest. Cells are our smallest level of organization. Together, those cells can form tissues. That tissue can form organs. Organs together can form organ systems. And then several organ systems put together, combined, creates an organism. When cells fuse and organize, they develop what's called emergent properties. These are functions and properties that individual cells are not capable of. For example, individual neurons are only capable of receiving and sending signals. But when they form a network, like our nervous system, it's able to produce something like consciousness and thought process. Once again, those are called emergent properties. In biology, scientists usually describe emergent properties by saying that the whole is greater than the sum of its parts. So we have those individual parts. Individual parts are arranged into a structure, and that creates the combination of those structures create emergent behavior, which gives us a much more complex life. Specialized tissues and organisms can develop by cell differentiation, which is the process of cells developing the ideal structures needed to carry out particular function. So what you always see when we talk about cells is pretty much an image that looks like this. This is the cell, but all cells, especially in the human body, don't look like this. They look much more complex or differentiated. You can see here we have this long elongated cell. Well, that's our nerve cell. We have red blood cells that have this kind of dip in the middle to increase surface area. We have these long, skinny muscle fiber cells. And within the cell, other things can differentiate, like the amount of mitochondria, or we look at plant cells and they have chloroplasts. But this idea of cell differentiation, specifically in the human body, is where we have our stem cell, and that stem cell has the capability to differentiate into other cells. Once it's at this level, though, they can't go back. So a stem cell can't become a nerve cell, which becomes then a stem cell again. Nerve cells become more nerve cells. There are actually 220 distinct types of specialized cells in the human body. They can form various types of stem cells. They form from various types of stem cells, and together that creates our tissue to form our organs. Okay. Once differentiated, cells can further fuse and mature to form specialized tissues. In this example here, cells specialize and fuse to create muscle fibers. Observable cell differentiation results from the expression of genes for tissue-specific proteins. This involves some parts of the genome being used while others are not. It is important to recall, though, that all cells in the bodies have identical genes, even if they're not being expressed. And remember, if you didn't cover this in middle school, that a gene is just a portion of the DNA. That's coding, of course, for a protein. Okay, a cell and organism's environment influences gene expression. Each cell of an organism expresses only a fraction of its genome, so their programming plays a key role in differentiation into cell types. So you can see here's our pre-fat cell transitioning, reprogramming into a fat cell. During development, changes in cell's environment, transcription factors influence which type differentiate into. Sequential changes in gene expression result in a series of physiological changes. Cell determination is the process through which an undifferentiated cell follows a development pathway. As a result of the environment and gene expression, its developmental fate is determined. Okay, so this is transcription factors. During development, the induction of transcription factors results in sequential gene expression. Recall that induction involves gene activation by chemical signals from other environment or cells. We talked a little bit about this idea of RNA polymerase, promoters, operators, when we were looking at positive and negative feedback mechanisms. So some of these concepts should sound a little familiar. Examples of the induction of transcription factors include homeotic genes, embryonic induction, seed germination, mutations causing abnormal development, genetic transplantation experiments, and regulation by microRNAs. Homeotic genes are genes that determine developmental patterns and sequences. They do so by programming various transcription factors that affect genes via regulatory pathways. For example, the Hox genes control the head-tail layout in embryos. In all bilateral organisms, the sequence of Hox genes expression ultimately determines the layout of the body. During development, embryonic cells are able to secrete signaling molecules that regulate transcription factors in neighboring cells. This induction ensures that cells differentiate and develop correctly. The timing and sequence of gene expression in embryos is essential for the proper development of cells into tissues and organs. The inducers in each cell's environment determines its development path. In plants, germination is the early growth of a seed that occurs when favorable conditions are met. It requires water, oxygen, and warmth to catalyze metabolic reactions. This is a germinating pea seed. The presence of these components in the environment results in a change in the seed's gene expression. The regulation and sequence of the genes results in the seed developing into a mature plant. Inherited genetic mutations can result in abnormal development. These types of mutations disrupt gene regulation pathways, which can cause malformations, deformities of the developing body. Mutations can also result in disorders like leukemia. Due to the alteration of genes in the immune system, stem cells are not able to properly differentiate into mature immune cells. So here we have our stem cell in healthy and leukemia. In healthy, it should differentiate into immune cells, but this is poorly differentiated immune cells. The link between gene expression and development has been supported by genetic transplantation experiments. These involve the insertion of genes into stem cells, which then become expressed. So right here it's showing the gene therapy that's occurring. Nuclear transfer is a form of cloning, where a nucleus is transplanted from a mature cell into an enucleated zygote. The zygote then develops by expressing the genes in the donated nucleus. Genetic regulation by microRNAs plays an important role in the development of organisms and the control of their cellular functions. MicroRNAs are a small piece of non-coding RNA that function in the post-transcriptional regulation of gene expression. They're typically about 22 nucleotides long and bind to mRNA to regulate translation. Okay, apoptosis is programmed cell death. That's its main definition here. That plays a role in the normal development and differentiation. It results from a series of biochemical events that can cause cell shrinkage, nuclear fragmentation, DNA fragmentation, or mRNA decay. Apoptosis can result from cell signaling and gene induction. During development, it's useful for creating borders and openings in the embryo's body. For example, separation of fingers and toes in developing embryos occurs because between the digits undergo apoptosis. This results in the formation of independent digits. If this apoptosis fails, or fingers remain joined as one, the toes or fingers remain joined as one and can be corrected with surgery. So apoptosis is actually helping this development of the body because it's causing this separation. Okay, during C. elegans development, 131 of 1,090 cells under apoptosis die under apoptosis. Knowledge of these reactions has helped us to understand mechanisms of apoptosis in other species. In order to conserve energy, flowers have a specific lifespan. In many species, chemicals released during pollination trigger apoptosis in the flower portion of the plant. So I'm going to go back to the slide just so you can make sure you have this written down. Apoptosis is just that programmed cell death that helps in development and differentiation.